Have you ever wondered about the complexities of stability testing in the pharmaceutical industry? It's a process that requires precision, time, and a significant amount of resources. But there's a way to streamline this process, thanks to the ICH Q1D Guideline. This guideline, entitled Bracketing and Matrixing Designs for Stability Testing of New Drug Substances and Products, provides a roadmap for optimizing stability testing protocols. It's all about using bracketing and matrixing designs. But what does this mean? Bracketing design is a method that involves testing a subset of samples from a larger group of similar samples. These samples are chosen based on criteria like formulation composition, container closure system, or the manufacturing process. The beauty of bracketing is that it allows stability data to be extrapolated to the untested samples within the same bracket. This reduces the number of samples required for testing. On the other hand, matrixing design involves testing a subset of samples at specific time points. This approach is based on the assumption that the stability of similar samples is expected to follow similar trends over time. Matrixing allows for the reduction of testing frequency while still providing sufficient data to assess stability characteristics reliably. The ICH Q1D Guideline also provides considerations for implementing these designs. It lays out criteria for selecting samples for testing, defining appropriate bracketing groups or matrices, establishing testing time points, and evaluating stability data. It underscores the importance of ensuring that the selected samples are representative of the entire population and that the chosen design approach is scientifically justified. But what about regulatory acceptance? The guideline recognizes that acceptance of these designs can vary among regulatory authorities and recommends consulting with regulatory agencies to ensure compliance with their specific requirements. However, many regulatory agencies accept and recognize the validity of these design approaches when appropriately justified and implemented. In a nutshell, the ICH Q1D Guideline is a valuable tool for streamlining stability testing protocols. By adopting bracketing and matrixing designs, pharmaceutical manufacturers can reduce the time, resources, and costs associated with stability testing. And the best part? They can still ensure the reliability and integrity of stability data for regulatory submission and approval. So the next time you ponder the challenges of stability testing, remember the ICH Q1D Guideline. It's a game changer, making stability testing in the pharmaceutical industry more efficient and manageable.